So now we're going to have a talk by Caine Barlow. And Caine's a fungal educator and mycologist. And in this talk he's going to be talking about psilocybin and its role as chemical ecology.
Yeah.
Yeah, I've got to turn the mic on.
Hello.
There we go.
Hello. Yes, that's it. I can hear my own voice, and that's not a good thing. Okay. Good evening. Okay, I'm going to begin with an acknowledgement of country. That I acknowledge the traditional custodians of the land which we're meeting on, the traditional lands of the Witchable Wayable people of the Bundjalung Nation. Pay my respects to custodians of the old ways, Morris leaders, and the knowledge holders of tomorrow. Sovereignty was never ceded, always was, always will be Aboriginal land. I also acknowledge the custodians of plant and fungi knowledge, the long lineage of knowledge holders whose wisdom has been passed down to us across time, space, and through culture. So tonight I'm talking about psilocybin in the context of a chemical ecology. So this is a little bit of a review. This is none of my own research, but this is just kind of a summary of the stuff that I've been going through. This is probably part one of a two-part talk, because essentially I've been diving down some pretty deep rabbit holes in terms of genetics, and this is kind of, it's a two-part story, because we're looking into full genomes at this point, which are like the entire genetic sequences of psilocybin. And we're now at the point where we've got, I think, close to a couple hundred full genome sequences, but mostly of Psilocybe cubensis and Psilocybe spirituosa. But we've been recently collecting a few more through other organisations. So the genus Psilocybe, we all know what psilocybe are. We're all going out foraging for these, I'm sure. I don't want you to dock yourselves. But essentially, psilocybe means bareheaded, and it refers to the separable gelatinous pellicle that covers the cap of the mushroom. So psilocybe are hygrophonous, and as this pellicle dries out, the cap changes colour. So like with Aeruginosa, for example, it changes colour from that beautiful caramel to then a light brown or a yellow, or in some cases even pure white. I found a patch at my university when I was in my late teens, and these mushrooms were completely white, and I think everybody missed them because they were just sort of white. The taxa psilocybe was originally named Agaricus tribus psilocybe by Elias Magnus Fries, and it was then elevated to genus by Paul Caine. Until 2009, the genus Psilocybe was synonymous with the genus Diconica. Diconica are quite commonly found on dung, and at a coarse level they can resemble some of the smaller little brown mushrooms type psilocybes. And so for a long time these two groups were considered the same. But then at some point there was some genetic work done, and a whole heap of researchers decided that the two groups need to be separated out, and then all the bluing species were then essentially moved into psilocybe. The type species for psilocybe was Psilocybe montana, and that is now Psilocybe Diconica montana. And as of 2009, the type species is now Psilocybe similanciata. So the type species being the species that we then refer back to as the species concept for what psilocybe is. I don't know, I was going to write another sentence, but I didn't finish. Sorry. this is that separable gelatinous pellicle. So this is the separable pellicle from Psilocybe semilanceata. Semilanceata, it's really easy to achieve this feature. The snap is, the cap is quite easy to snap, and yeah, you can see that kind of that layer. This is the same feature on the Psilocybe cubensis. It's a little bit harder. I had to ruin quite a number of mushrooms to get this example. This is the global distribution of Psilocybe according to Wikipedia. This map is quite out of date. There are now a lot more species known, and the distribution is quite different. So I have included something a little bit more up to date. And these are all the iNaturalist records currently mapped out. Do you guys want to move further down the front?
No, we can't quite seem to find a good angle.
Oh, well, camera on this side. Front row's good.
I'll take you. Peering like meerkats at the back.
But you can see the density from the Wikipedia map is really markedly different. And the beauty of iNaturalist is you can dive down into this data a little bit more and, you know, kind of look at what species are in which regions, and even kind of go through what species are present within a given region when you zoom in. So we have, I guess, probably 4 kind of areas of endemism. So we've got the kind of Central America area where there's about 33 species. Europe where there's 15 species. Oceania, so Australia and New Zealand where we've got currently 18 different species listed. And I guess another point on this is that these are not all necessarily species that are native to these areas. Some of these are introduced. So, and I'm going to talk about more about that tomorrow in my other talk. And then we have Southeast Asia, which has 14 species, but I have a strong suspicion that there's a hell of a lot more in that area. My own particular theory is that the centre of origin of Psilocybe is quite possibly Southeast Asia. But tonight, I'm talking about psilocybin.
Yeah, psilocybin.
Yay, psilocybin. I like psilocybin. It's a cool compound.
Are you talking about psilocin?
Well, that's an interesting point. When we talk about psilocybin, we are essentially also talking about psilocin, because psilocybin is the prodrug to psilocin.
Is the what?
It's the prodrug.
Prodrug.
What does that mean?
What does that mean? That means that psilocybin is kind of like the holding state within the mushroom. It's really what the mushroom wants to have. It wants to produce lots of psilocybin. Because then the psilocin breaks down really quite readily. As soon as it's dephosphorylated, the phosphor, and I'm going to come back to this though, as soon as the phosphor group is removed, the psilocin is oxidized really quite rapidly and breaks down.
And that's what the fluid is, right?
Well, and I will come back to this point. But yeah, it breaks down rapidly. It doesn't last very long. Yeah, the mushroom has particular ecological uses for this compound, so it kind of wants to make sure that it's in a state that it doesn't break down too quickly. But it's psilocin that it's the psilocin oxidization that causes that fluid reaction in the flesh of the mushroom. Psilocybin emerged as a genus approximately 65 million years ago as a wood decomposer. It emerged as a genus after the KPG extinction event, the meteorite that hit Earth around Central America, I suppose.
You think that's where it came from?
No.
Didn't come from aliens from outer space?
No, I don't think so. My own theory is that I think psilocybin was around a lot longer before this point. The thing is that to get these dates, we have to use either fossil records. So like in plants, you have to use fossil records to try to approximately date some of these evolutionary points. In the case with fungi, it's fungi that have been found in amber. So this particular date is based on a sample of Agaricus praecox that was found in amber. And they've dated that to about 75 million years ago. And so through using it as an outgroup in phylogenetic trees, so they've then been able to date these things. Again, this is more of my talk I'm going to be talking about tomorrow.
Oh, what time?
11:30, I think.
Oh, that's early, I'll try.
11:30?
4 minutes.
So 11. Okay, thank you.
It's early, but I'll try.
So 65 million years ago is a long time. It's a long time, and psilocybin has diverged really quite significantly. And they have adapted to soil, to dung, so you've got Psilocybin cubensis. So the soil species being kind of like section Zapatecora. So Psilocybin orglandiae, for example, which have recently been found here in Australia. Humus, which is kind of really litter-rich soil, so there's lots of plant material that's not fully decomposed within the soil. So the mycelium is kind of growing through that, and with lots of moisture, the fungi fruit. I haven't included the entire illustration here. So this is from the Alexander Bradshaw paper that was published last year. But it does illustrate how some of these species have adapted to different ecological niches. So soil and wood. The dung-loving species are here. But this phylogenetic tree, so this is a map of the evolution of psilocybin. But in some cases it does include other species because the psilocybin is produced by the psilocybin gene cluster, and at certain points in the evolution of psilocybin, it's been sharing its psilocybin gene cluster with other species of mushrooms, other genera of mushrooms. So Paniolus, for example, the blooming Paniolus-cyanesis, Gymnopatus, which are also wood-loving species.
Oh, okay, so you found them. Okay, there you go.
It's still quite speculative, but it's hypothesized that the byproducts of psilocybin, so psilocybin to psilocin, and then to this kind of structure. So you can see psilocin kind of here in this, and it's bonded to another psilocin. And this is referred to as a quinoid coupled dimer. So these things kind of form a matrix of, like a crystalline matrix. And it's been speculated that they infer damage to insects and gastropods when they consume mushrooms. So they eat the mushroom, it starts to kind of gouge out their intestinal tract, so they stop eating and move on. But it's also been speculated as part of that process that then it's the insects and gastropods that also then help spread spores as well. So there's still a lot of teasing this out ecologically. It's also been speculated that psilocybin is potentially a good UV protector. as a result of these same crystalline structures.
Officer, it's for my skin.
And as I mentioned, some ecologies have allowed for horizontal gene transfer for bacteria to essentially, by inhabiting the same space, so Panaeolus living in the same dung as Psilocybe, bacteria are able to carry some parts of the DNA into other species, and at some point, well, the psilocybin gene cluster is clearly very beneficial to psilocybe. It survived as a genus for 65 million years. There are species growing over the entire planet. We've got 165 species. Another aspect of this that's quite interesting is woodlover paralysis. And I have a question for you, the audience, is when foraging for mushrooms, do you ever find disabled slugs? Because I do wonder if perhaps woodlover paralysis acts on some of the insects or slugs to also then inhibit their behaviour and stop predation. Psilocybin is a secondary metabolite that's formed by biosynthesis from L-tryptophan. L-tryptophan is a very, very common amino acid. It's probably accessed either directly from the environment or from the breakdown of the food products that the fungi are breaking down. It's important to point out that potency is mostly a reflection of physiology. And specifically resource allocation. We see this in many of the species of cubensis, where, and other species as well, but particularly with cubensis, I think, because it's so commonly grown, and there are so many different mutants of this species, we're in a position where we can start to compare the different physiologies and see and then compare potencies. Part of this is the fungi are dependent on how much nutrient they can access. It's really well known, particularly with Psilocybe seborrheirosa, that wild grown seborrheirosa are relatively weak, but the wood chip ones are much more potent. And I have anecdotal knowledge of people who've grown them indoors on really, really dense substrates that they get really strong, like super strong. But this is also seen in some of the literature. There's literature that shows, and I think it was Josh and Gart and a couple of other people who've compared cubensis grown from dung out in fields to then cultivated, and the difference is fourfold, in some cases to like 10 times as strong. And then there's also other aspects of how the resource allocation works with what substrates you give. So there was the example of maintaining substrate. So if you've grown your cubensis on one substrate, it's best to maintain that substrate when you bulk it out, and that increases the potency of those mushrooms. They're already trained within that breakdown, those enzymes that they're using to break down the substrate. So changing it up means that they then have to change up their metabolism. There's also the leucistic and albino traits that are associated with potency increases relative to other cubensis lineages within the same populations. So, and this is thought to be with the mutation in the melatonin pathway, that the L-tryptophan is not being processed to then create those compounds, and is therefore being redirected into the psilocybin pathway, and hence resulting in far more potent mushrooms. Another aspect of this that's quite interesting at the moment is how psilocybin is being treated in a human ecology. We have some fairly significant underground and above ground market forces happening at the moment. What was seen as a recreational drug 10 years ago or 15 years ago for the spiritual development, which have all now been classified as non-clinical use, is now an emerging mental health treatment. So there's a lot of research, there's a lot of time and effort being pushed into how to better grow mushrooms to then be able to extract psilocybin, and then also the mental health programs that are being developed around that. And this is the consequence, in part, because of changes in legislation as well. Overseas, here in Australia, we now have this situation where psilocybin can be descheduled to Schedule 8 for very, very particular mental health situations. And then there's also decriminalisation, which is allowing a lot of really fascinating research, particularly with groups like Pike and the psilocybin cut groups who are legally allowed to grow mushrooms and legally allowed to then do the subsequent chemical analysis of these things. There's a hell of a lot of commercial interest, both above ground and below ground. There's also a lot to be gained from spurious claims. There are people coming up with, all these different varieties of cubensis and claims to be breeding different species, stronger species. There's a whole lot of interest in exotics from different parts of the world. Particularly things like that are coming out of New Zealand at the moment. I might wait for the big to pass and just drink water. Okay. But yeah, there is a lot of interest at the moment in the exotic species, particularly section Zapaticorum, sections Severogenosa, which Psilocybe severogenosa is within, have this problem with wood lover paralysis. So people are kind of not so interested in exploring that group. They're more interested in looking at, some of the Mexican exotics, section Zapaticorum. There's currently a mushroom getting around referred to as JIYP that I think is something like psilocybin, which is an insane amount. Compared to your 1% that Psilocybe cubensis produce, that's a marked increase. So we're starting to see an end of this cubensis-centric kind of Psilocybe ecology that we have. I'm now going to start talking about the psilocybin gene cluster. Psilocybin is biosynthesized by this a group of enzymes encoded within a really highly conserved gene region, referred to as the psilocybin gene cluster. So essentially, a group of enzymes, we have Psi-D, Psi-K, Psi-H, and Psi-M. And I'm going to go into this in a little bit more detail. It's highly conserved, meaning that it's barely changed. It barely changes over time. So any kind of deleterious mutations mean probably, those mutations are not going to be carried forward. Small changes within those genes are okay, and we can actually create phylogenetic trees to look how these sections have evolved over time. But they haven't evolved enough to be able to really change how these enzymes functionally work. This has resulted then in the fact that, psilocybe have this massive global distribution to approximately 165 species, distributed across numerous ecologies. It's a fascinating protective mechanism. And the fact that it evolved 65 million years ago and is overall mostly the same with some variations of the gene cluster. But there are variations in how the genes are organised within the different clades and sections, and there have been double ups, which kind of have some interesting consequences, and that probably needs to be explored in more detail. Again, and this point of how beneficial this compound is allowed this horizontal gene transfer to occur. This was the first proposed version. So we start with, in this version, we started with L-tryptophan, the Psi-D enzyme, and decarboxylated to tryptamine. The Psi-M enzyme then methylated that to dimethyltryptamine. Psi-H oxidized it to psilocin, and then Psi-K then did this phosphorylation to psilocybin. There was a really cool paper that came out, and this is just to explain what these different Psi-D, Psi-M, Psi-D, it's kind of a little bit meaningless. So I'm move further over here. You good? Yeah. These are all bits of mouthfuls, but and I'm essentially just going to explain them as I kind of walk through. But as I said, the psilocybin gene cluster has been organised in different ways within the different parts of the genus Psilocybin. So clade 1 and clade 2. These are some fairly major changes in the order, but it doesn't really matter. It doesn't matter how it's ordered, it still does the same thing. When I say clade 1 and clade 2, so this is a full phylogenetic tree. I'm sorry, it's so big it's all going to be blurred and meaningless at this point, but I'm going to go into more detail tomorrow. But at some point about 55 million years ago, the whole genus Psilocybe kind of diverged, and you've got these two different groups. Yeah, kind of like cousins. Yeah, it's a bit more complicated than that, but it's generally different variations of the same. Yeah. But this kind of adds a little bit more, I'm showing this because it adds a little bit more detail to what I'm going to demonstrate in a few more slides. But this is the currently accepted version. So this is adapted from the Frick et al. Paper from 2017. And if you want to read more about this, I highly recommend this paper. It's a good read. But we start with L-tryptophan. We have Psi-D that decarboxylates the tryptophan, so it removes this particular group here and just drops a hydrogen here. So we just then have tryptamine. Psi-H then adds a hydroxyl group to this part of the tryptamine compound, and then we have Psi-K to add the phosphor group, and that forms an all-beta system. And then things get a little bit more interesting. So from Nor-Bayer system, we have Psi-M, which methylates the nitrogen and adds a single methyl group. You then have another Psi-M come along and it adds an extra methyl group, and that produces psilocybin. It can even go a step further, and I point this out because I'll come back to this later. It can add in a third methyl group to form Entheogenesis. But also, since we now have this phosphor group here, you can have dephosphorylation. So dephosphorylation of beta system then produces Nor-psilocin. Nor-psilocin is not hallucinogenic, but it has been shown to have really strong antidepressant effects. So this is a really interesting compound in mental health. The dephosphorylation from psilocybin, of course, results in the psilocin. And with Entheogenesis, it results in 4-HOTMT, which is a really, really interesting compound, and I'll come back to it. So 2024 was a fantastic year in psilocybin research. We had the Bradshaw paper, Phylogenomics of Psychoactive Mushroom Genus Psilocybin and Evolution of the Psilocybin Biosynthetic Gene Cluster. This paper was fantastic, and I'll show it in a bit more detail in a moment. There was also then the long-awaited McTaggart paper, demonstrating that wood-loving mushrooms from Australia are saprotrophic invaders in the northern hemisphere. So this was the paper that demonstrated that Psilocybe cubensis, Psilocybe cyanesum, Psilocybe azurescens, quite likely Psilocybe elenii are just Suberoganosa. They're just an introduced species.
You're on that paper, right?
I beg pardon?
You're an author on that paper.
I am.
Pull that out.
Thank you. But there was also some other really cool stuff that came out of that paper that never ended up in the final peer-reviewed paper. So I recommend going and finding the preprint. And I'm going to bring that up later on as well.
What's the saprotrophic?
Sorry?
What is saprotrophic? That's wood.
Preprint.
It's saprotrophic.
Saprotrophic.
Yeah, thank you.
What is it?
Saprotrophic, what is it? It's wood decomposing. Yeah. So I guess grammatically it's a bit of a weird title because we're using wood-loving, wood decomposing, which is worded in a different way. Yeah. There was also another Bradshaw paper that came out talking about a new species of magic mushroom in Africa. And I'm going to talk about that tomorrow because there was some cool stuff that came out of that paper. I'm not really going to cover that one tonight. And then there was also the Bollinger 2024 paper. Well, it wasn't really so much a paper, but it's just a communication or announcement. But it was announcing a group of really high quality draft genomes of ecologically and geographically diverse psilocybe species. Which has kind of helped to further illustrate stuff about the psilocybin gene cluster. So this is essentially the title section of the Bradshaw paper. And this is the most, I think probably the most notable figure within the paper. It gives the phylogenetic tree of all the holotypes that the Bradshaw group were able to access. And they genome sequenced all these holotypes. Holotypes being the species that the sample that species was then named and described from. What they've done is then added the order of and direction, which is also kind of interesting, of where like the side D, the side K, side H, and side M sit within the gene cluster. And as you can see, there's a fair bit of variation going on between some of these. And then this is also the subsequent phylogenetic tree that was made out of then just comparing all those particular genes themselves, so the side D gene, the side K, side M, side H. It's probably mostly meaningless because it's all blurry. But if you're interested in this, again, I recommend reading this paper. It's really quite interesting. This is the title of the Bollinger paper.
You're also on. Are you also on?
Yes, I am. I'm not promoting myself, I promise. This was the original version then of how the gene clusters aligned. So this is section Zapatacorum. So what do we have? We have psi N, that's a transport gene, psi H, and in this case there are two psi H's, which is kind of interesting. We then have the psi K and the psi D. And so this is the clade 1 group, and these are kind of mostly the same. So that clade 1 group is kind of, so section Zapatacorum, Psilocybin, section Psilocybin Zapatacorum, Psilocybin mexicana as the representative of section mexicana. We then have Seberogenosa. And as you can see, this is it's fairly different. Things have moved around a fair bit, and this is a good example of clade 2. So what have we got? The transport genes are in the same place. The psi H's are kind of mostly in the same place. These have swapped around a bit, so the psi D has moved over here. But also we have a third psi H, which is interesting. Why is this group triplicating the psi H? We don't know yet. But then, as you can see, Psilocybe cervica, which is also in clade 2, it's kind of aligned slightly differently again, and closer to cubensis. And then these are some of the ones that have been sequenced from horizontal gene transfer. So you've got the same genes, again, ordered in a different way. This is a much better example. It kind of illustrates how, again, you've got section Zapatacorum, section Semilantiate, and section Mexicanae. These are all mostly the same. Psilocybe gandalfiana, which is a new species that is, I think, about to be published, is fairly much the same as then those other species there. So Psilocybe carolina, which I think is section as Tacorum, and section Psilocybe cervica. This is Psilocybe cubensis penis envy, and it's exactly the same as Psilocybe cubensis from Queensland. Like there's no difference in that gene cluster. Here we have Seberogenosa, Eleni, and Azurisums. These three have been shown to be the same species. Again, as you'd expect, they're mostly the same. You've only got a few gaps, I think, in the Seberogenosa that are kind of interesting, and I know people are wanting to dive more into those gaps, but it's a matter of working out what genes could be in those spaces. There may be genes, or it just might be filler. We don't know. Pardon?
There's no such thing as filler.
No such thing as filler. Okay. Well, notably, when we were working on this paper, Ian was kind of like, why is there this massive space here? And dived in and then found that, yeah, it was a hypothetical pi-R gene. So, likely right, there's probably other things here. It's just a matter of blasting them, working out what they are. Pseudo genes. Yep, that's what I meant by filler. It was kind of interesting because Science picked this paper up and kind of then made this statement about how 50 species in the Psilocybe genus, out of 50 species, 35 had a novel gene arrangement. Yeah, they might be novel generations, but they don't necessarily mean that there's going to be any difference in the biosynthesis. So, for some reason, there's kind of like this push to go, there's this space here where you can explore all these new versions of these tryptamines. Hang on, no, they're the same tryptamines, they're being produced in the same way. And as part of this narrative around, I think, this kind of commercial exploitation of philosophy and other compounds that may be present. Well, that's certainly how I read it. I don't know, you may all feel differently on that.
Could be a diversity issue.
Well, I don't know. I'm a big believer in a cube kind of thing. So, it's a consequence of diversity. But I keep coming back to this kind of narrative of like, well, you still end up with just having psilocybin or north psilocin. Because it's all enzyme driven. It can only be, that cluster can only produce these compounds in that particular way. So, and going to that Frick paper, it talks about like the different substrates, how they used some of the tryptamines as substrates to demonstrate, it can only work this particular way. It can't work in other orders. So I come back to this again. So yeah, as you said, there is a diversity, but it's still producing fairly similar, the same compounds. They're just ordered in a different way. It's just over time, with speciation and copying, things have been shunted around a little bit. The end product is still the same. Now why did I throw this in? I threw this in because yes, this is my cube for cube is a cube. This has become a bit of a question. It's become a point of debate over, I think, around about 20 years. Perhaps the entire lifetime of the shroomery, for example, is people go on about, oh, look at my new cube, that cube, this thing that I bred. And then you get the other people, and I'm one of those. Is a cube? And I think, yes, it is a cube. There are plenty of websites that talk about how different strains of cubensis, the Amazonian, Cambodian, Ecuador, Gulf Coast, Papua, etc., have a recorded difference in effect. So you might take Amazonian, they might say it produces a really blissful kind of dreamy effect, whereas penis envy might produce a really dark kind of spiritual effect, or I'm making this up. But you can go to some of these sites that describe these different varieties, these cultivars of cubensis, and they will go into detail about the different effects that the different strains produce. Alistair McTaggart and the rest of his group in 2023 published a paper on cubensis, and that's where this figure comes from. And this kind of shows all the different cultivated varieties compared to the Queensland cubensis. Essentially illustrating that that there's been a bit of a diversity from the natural populations to then all of the inbred varieties that have been passed around. But the paper reports that there is essentially no difference in the psilocybin gene cluster across the whole cubensis group. And that essentially changes in the levels of psilocybin or psilocin as simply just the result of physiology. It's the substrate that a mushroom is grown on. or some of the mutations. So, and I guess kind of how some of the different pathways are then moving the other compounds around. So like the L-tryptophan. So the broken melatonin pathway, for example, means that L-tryptophan is not being used in that pathway and is moved somewhere else. So into the psilocybin pathway. The paper then also shows there's a low diversity in naturalized and cultivation populations of cubensis compared to the high genetic diversity of all examined alleles in Spirogynosa, as an example. Spirogynosa has a lot of diversity, and I'm going to show that shortly. That's not to say that there might not be other genes outside the psilocybin gene cluster that we're unaware of at this point too. So there may be things doing other interesting things. We know that cubensis produce harmalines, but then also, what is it? Sorry, I'm having a blank. Terpenes. Yes, thank you. Why mushrooms are producing terpenes for, I guess, their own reasons, but it's kind of, all the cubensis people kind of were like really excited by, there are terpenes in magic mushrooms. But how does that then, is there an entourage effect, etc., the kind of conversations that come out of that space? And why are there harmalines? But the thing is with some of these other compounds that are being produced, particularly like the harmalines, they're not producing enough harmaline compounds to change the effect of the psilocybin or psilocin that you're then consuming.
So harmalines are the ones in the fly agaricus?
Harmalines are the compound, they're MAO inhibitors. So like when you take ayahuasca, for example, you're taking DMT with a harmaline. And it's the harmaline that deactivates the compound in your digestive system that would ordinarily break down the DMT.
Is that similar to an MAO inhibitor?
Yeah.
Prolongs the action. Prolongs the action.
Yeah, it prolongs the action. But it stops the degradation as well. So that it may be some aspect of this protective part of trying to maintain high levels of the psilocybin for the protective benefits of the mushrooms. This is the McTaggart paper. And since I've asked, is a cube a cube, it's only fair to ask, is a sub a sub? There is lots of diversity in the psilocybin gene cluster in Spirogynosa itself. The main genes are all in the same locations, but you've got a lot of movement of things being moved around. So these CIH is being shifted around. You've got triplicates in some and not in others. So what's going on in Spirogynosa? And then the other interesting thing is section Spirogynosa produces wood-level paralysis as well. There is some speculation that maybe these triplicated CIHs have played some part in this. Because it seems to be that this particular group, in Clade 2, the Spirogynosa group, have this really messed up kind of psilocybin gene cluster that potentially there's other things going on in there that we're not aware of. And it's an area of interest. In a yeast model of the biosynthesis of psilocybin, so taking the psilocybin gene cluster and inserting it into yeasts and then giving it a substrate, and then having multiple copies of cyan has resulted in a 21-fold increase of the amount of 4-HOTMT, which is that other one that I demonstrated a whole bunch of slides ago. So this one here. So you've got this extra cyan that produces this third methyl group, and then that's dephosphorylated to 4-HOTMT. which is then kind of interesting. Yeah, so this huge increase in 4-HO-TNT, but we're not seeing extra cyans in this example here. So, don't know. So I guess the question is then, are there genes that we're not seeing? And the answer is quite possibly. And this is just to kind of complement the wood level paralysis thing, where for those who aren't aware of wood level paralysis, who hasn't heard of wood level paralysis? You haven't. Okay, you haven't either. Cool. Wood level paralysis is a particularly interesting effect that happens in some Psilocybin aeruginosa, where it produces a temporary paralysis of the body. So you take a mushroom, and within a couple of hours you may start to notice that you can't use like your hands, you may not be able to walk. In more severe cases, eating is a problem.
I have heard of it actually, yeah.
Yeah, okay. And in really severe cases, it even affects your breathing. So, yeah, there are potentially deaths related to this. So, it's something that we're kind of trying to raise a little bit of attention about it, so people are aware that the possibility is there. But there's also, from this whole kind of biosynthesis narrative of, well, where in the mushroom's genome is it happening? And we're fairly certain because, well, we did a survey a few years ago, and from the results, it's fairly clear that it's something in the mushroom itself, and it's not some kind of other environmental toxin.
Does it affect everyone the same? Or?
Sorry?
Does it affect everyone the same?
No. There seems to be a little bit of kind of a personal thing about it as well. But, there's a... I mean, yeah.
There's no defining features like the unworked out.
I guess there are defining features of it. You're not able to move.
I mean, not the person.
Oh no, we did look at that as well. You know, we asked people to report allergies and like that, and across the board, it didn't really seem to matter.
Caine, have we identified the actual chemical that causes the paralysis?
No, we have not.
That's not vecuronium or...
No, so this is all speculative kind of talk at the moment about what's the actual compound. But, and this is what I'm trying to demonstrate on the slide, is we know from bufo species which have bufotenine with
N-methylation.
N-methylation, thank you, Simon. It results in bufotenidine, which is known to also cause a paralysis effect. So we're drawing an equivalence between the bufotenidine and then the 4-hydroxy-TNT, 4-HO-TNT, as maybe either the guilty compound itself or the precursor to that guilty compound. And there has been some suggestion within the community that in the same way that psilocin forms these kind of matrix dimer compounds, that maybe the 4-HO-TNT that does something similar, that creates some kind of compound that then slots into a receptor temporarily in order to cause that paralysis. But again, this is complete speculation.
I've seen people having like kind of uncontrolled like their muscles and things like that, but it was only for like a short 30-second or a minute.
Oh, okay. That sounds more like twitching or...
No, it wasn't twitching, they were fully like losing control. Almost passing out.
Right. Simon, do you have any comments on that? Or?
No, I think like the paralysis usually comes sort of on and off in waves across the period of the trip, rather than being like a really short, quick, shock and weakness.
that's the end of my talk. So, question time.
We have time for a few questions.
Yeah.
And I guess I'm going to take a liberty and start myself. So, you know, look at all these gene clusters. When we're looking at all these gene clusters, you know, I think that there's been a lot of focus on them as a cluster themselves and their, you know, their arrangement. And that mainly comes from when they were originally cloned and put into yeast to manufacture psilocybin. So my feeling on all this is that the organisation of those genes will mean diddly squat, because the regulatory regions above them are still there, and all those genes are regulated by factors outside of that gene cluster. So, is anyone looking at the transcriptor factor binding sites above those genes and then trying to work out which of those genes are binding on each promoter of these genes, and then trying to do some kind of like quantitative analysis on how each of those, how highly they're expressed?
So you're talking about in the cases of where they're rearranged in different species, between species?
More just between species, and look at their binding factors on their promoters, identifying which transcription factors are upregulating these genes, and then looking at the levels of those transcription factors in fruit antibodies. That would give you an idea of.
Oh, look, I agree. I think there's a whole area of research here in terms of, you know, and it kind of comes into this entourage effect of, you know, like how these different tryptamines are being produced and what amounts over what time period. So, yeah, at any given time, there's a flux going on within how many of those compounds are available. I think definitely with the Asparaginose group, that's really interesting, and I would really love to be able to dive further into that. And I guess that's like kind of a PhD level thing of diving in and going, okay, what's filling those spaces? How are these being? You know, look, it's my feeling that different populations of subs are producing different things, and the effects are different compared to the commensals that do seem to be fairly consistent.
If only we knew an expert in genetics and genetic regulation.
There's lots of research to be done. All right, is there any other questions?
Maybe I'm being stupid, but doesn't everything organic have aromatic molecules? And terpenes.
Aromatic?
Everything organic has aromatic molecules there.
Oh God, yes.
So what you seem like, you said before that it was sort of like a surprise to find them in mushrooms. Why was it a surprise? Terpenes.
Terpenes?
Yeah.
I think just because nobody had been looking for them. It was just like someone had just kind of published this paper of like, oh, there are terpenes within psilocybin. I think there are a variety of psilocybins. So, and it was definitely, certainly the group of people I kind of worked with, was the convincing, the cannabis people going, oh wow, you know, like entourage effect. You know, like these terpenes are contributing to this, potentially contributing to this psychedelic effect. You know, but
It had been kind of overlooked a little bit.
it had been overlooked. I think that's where the surprise was. Yeah, people wanting to make claims about the potential additional effect that the terpenes may have. I mean, it's kind of speculative, I feel. No, look, it's a further area of research. It's like, they hadn't been looking for them, someone found them and published a paper. Like, oh, okay. Terpenes have been found in other species, like Entheogenesis, for example, and a few other things. But just at that point, not in the psilocybins.
Well, if we haven't got any more questions, or if we have, we'll wrap it up.
Can we catch Caine anywhere?
Yes, we can.
Oh!
I thought we were going to do a spiel for it.
Who's doing this for you? You or me?
You might as well do it. You're standing there with the projector.
Alright, okay then. This tent here has been set up in conjunction between the MardiGrass group and also Entheogenesis Australis. Do you have flyers, by the way?
There are flyers
down,
on the top there.
Down here, there are flyers here. But Entheogenesis Australis is holding a symposium, a 4-day, 3-night symposium at the end of the year in November, where all of the talks that have been happening here, similar talks, are going to happen at that symposium over those whole 4 days. So lots of opportunities to talk about fungi and other plant medicines, your cacti, acacias, etc. Psychedelic culture, all talks about psychedelic therapy, etc. Also keep an eye out for the EGA journal. So the EGA is publishing a journal this year, and that will be available within the next, hopefully, few months, not too much longer. And we're featuring articles by Graham St. John, It's been a long day today. Des Stranaki and Anna Cobra. We've got lots of cool Australian psychedelic art as well, illustrating the book. But yes, applications are also now open for talks. And tickets will be available soon. Already up. Look at that. So if this is kind of the thing you're interested in, I highly recommend coming along to Garden States at the end of the year.
Cool. I'll just
and then some other kind of groups in Australia that you might be interested in if you not aware.
Don't worry, Caine, I'm going to tilt you up as soon as I've done this. Okay, let's start.
Okay, everybody. Great to see you all. Thanks for making time to come this morning for this session. The next 3 sessions here in the tent will be back towards the psychedelic sort of subject, and we'll be starting with Caine Barlow here. Caine has the acknowledgement up here, so I will leave it to him to provide the acknowledgement of country. But I have to say that it's really great to be back on Bunjalung country, as I do each year now around this time of the year. So it's really it's such a comfortable and lovely place to be. And I would like to pay my own respects to elders, past and present, here in Bunjalung country. So our first talk today will be by Caine Barlow. He's a fungi educator, mycologist based in Melbourne, as a few of us are. He started foraging mushrooms back in the early 1990s and started cultivating gourmet fungi in the mid-2000s. So he has a lot of form in this area. He's been teaching mushroom cultivation in person over the last 8 or so years. In addition to fungi, he's had a long-term interest in ethnobotany more generally, ethnobotanical literature, and growing medicinal plants. So the topic for his talk today is psilocybin, using barcoding genes to better understand the global ecology. So thanks very much, Caine.
Thank you, Mark. Thank you, Mark. Good morning, everybody. Okay, sorry about the slides, they're a little bit washed out. But this is quite a visual presentation. Acknowledgement of country. I'd like to acknowledge the traditional custodians of the land that we're meeting on this morning. The traditional land of the Widjabul Waibel people of the Bunjalung Nation. I pay my respects to the custodians of the old ways, today's leaders, and the knowledge holders of tomorrow. Sovereignty was never ceded, always was, always will be Aboriginal land. I also like to acknowledge the custodians of plant and fungi knowledge, the long lineage of knowledge holders, whose wisdom has been passed down to us across time, space, and through culture. So So this morning I'm talking about psilocybin, and I'm talking about barcoding genes. There is an incredible amount of really good, high quality genetic information that's hitting the internet, and I think impressively it's all citizen science, or mostly all citizen scientists who are contributing to this growing narrative. And psilocybin is part of this picture. There are people who are out foraging multiple genera and then getting them sequenced, and all that data is landing on the internet, and you can access it and then process it in your own way. So this is what I've been looking at for the last year. This is my little attempt at humour this morning. This is essentially 250 ITS sequences from psilocybin from across the planet. And I've been doing this the hard way. I've been manually curating this and aligning it in order to try to develop a narrative and a better understanding of their global ecology. I'm going to come back to this slide in a bit more detail later. I'll just cover this very briefly, but essentially psilocybin means bareheaded, and that refers to the several gelatinous pellicle that covers the top of the cap. So psilocybin are hygrophonous, which means they change colour as they dry out. So this pellicle, as it dries, like with Psilocybin sphaerognosa is a great example, has a beautiful caramel coloured cap. As it dries out, it kind of goes to tan or light brown, and in some cases yellow, or in other cases even white. The taxa was originally named Agaricus tribus Psilocybi by Elias Magnus Fries, and that was then elevated to genus by Paul Caine. Until 2009, the genus Psilocybi was synonymous with the genus Diconica. The type species was Psilocybi montana, which is now Diconica montana, and as of 2009, the type species is Psilocybi semilanceata. This is an illustration of the gelatinous pellicle.
Wow.
Oops. And you can kind of see it here. You can snap the cap, peel it away, and you'll get that. You should be able to find that later. It's quite easy to do with semilanceata. It's a little bit more difficult with cubensis or sphaerognosa. So this is the same thing on cubensis. This is a little bit harder to achieve. I had to ruin quite a few mushrooms to get this. But you can see it. You can see it here. Uh, 2024 was a fantastic year for psilocybi research. We had some fairly significant papers come out last year, notably the Bradshaw et al. Paper, the long-awaited McTaggart paper that demonstrated that our wood-loving mushrooms from Australia have travelled overseas and have hence been named other species. There was also another Bradshaw paper that came out at the beginning of December that's contributed a whole heap of stuff as well, but that was a preprint. It hasn't been peer-reviewed or published formally, but it was still a worthwhile read. And then a Bollinger paper that was published just shortly after Christmas, so everyone kind of missed it. This is the title page of the Bradshaw paper, and this is probably the most notable figure from that paper. And what we have here is a phylogenetic tree demonstrating the predicted evolution of psilocybi. And I'm going to go into this in a little bit of detail over the next few slides. But it covered quite a few details. It also covered the psilocybin gene cluster, and if you were here last night, I went into that in a lot of detail. So
This group also did phylogenetic trees of the various genes of the psilocybin gene cluster as well. It's a great read, highly recommend it. But one of the notable things about this paper was that it confirmed some of the results from the Ramirez-Cruz paper in 2013, showing that psilocybe diverged at some point, and you've got two large clades. So you've got these two large groups, and looking at the psilocybin gene cluster kind of confirms how that divergence goes, the effects of that divergence. It also confirmed some of the sections as well. So sections Cordysphorae, Mexicanae, Cubensae. For some reason though, both papers kind of ignore section Sphaeroganosa. Then there was the McTaggart paper, where this group kind of demonstrated that psilocybe cyanescens, psilocybe azurescens, and quite likely psilocybe allenii are species that come from Australia, which has started a fair bit of debate on the internet. This is the Bollinger paper, and this is the psilocybin gene cluster that figure from that paper. So again, talking about like the two different clades. So you've got the clade 1. This is how the psilocybin gene cluster is aligned within that clade, and then the variations within clade 2, but also the variations between the different sections. So you've got section Cubensae here, Aztecorum, and then section Sphaeroganosae, which is doing its own funky thing. And there's a lot of questions that come out of why is section Sphaeroganosa doing what it is doing. Then we had the Bradshaw preprint, and this is a really lovely figure that comes from that. But again, demonstrating that we have these two clades, so clade 1 and clade 2, and then the divergence of all the different sections. So these are probably the most, well, I feel anyway, the three most notable species. So Psilocybe cubensis, Psilocybe sphaeroganosa, and Psilocybe semilanceata. So your gold tops, by virtue of Psilocybe cyanescens being part of section Sphaeroganosa, your wavy caps, and then your liberty caps. So psilocybe emerged approximately 65 million years ago as a wood decomposing fungus. We're not quite sure where it emerged. But by starting to dive into the genetics, we should be able to work out at least a number of narratives of what occurred. But over that time period, they have evolved and adapted to a number of different ecologies. So soil, dung, humus, in addition to the wood. What's notable about this whole genus is just the highly conserved psilocybin gene cluster. It has been rearranged through various speciation events, but it's still present and proves to be highly beneficial for this genus. This is the global distribution of psilocybe according to Wikipedia. This illustration is a little bit out of date. It's okay, it covers the main areas. But this is the current map. It's a little bit washed out, I assume. Is it okay? You can read it? Yeah. We have about 4 areas of endemism, but also keeping in mind that some of the species that turn up in these areas are introduced into that region. But what's been quite notable is the number of species in Central America. So approximately 33 species, approximately 15 species in Europe, and 18 species. There are a whole bunch of others. There is kind of this area up here. Oops. There's this area up here, but this is mostly the section Subaruginosae group, which have travelled from Oceania, and Oceania has about approximately 18 species. And then we have Southeast Asia, where there's currently 14 species, but I have a prediction that there's a hell of a lot more there. Being a naturalist, this has only had observations of things that have been found recently. So there are approximately 165 species, but some of those have not been found recently. So with not having observations, they don't turn up on these maps. These are the species that are present in Australia. So Psilocybe subaruginosa. We have Psilocybe cubensis, but that's an introduced species, hence the cross. We then have Psilocybe ludicea, Psilocybe papuana, Libertycaps, Zimbuiensis, Tasmaniana, Aucklandiae, Cordysphorae. There was this weird Psilocybe aztecorum, but then when a few of us went and looked at this in a bit more detail, it turned out to be incorrect. It was something completely different, not even Psilocybe. And just to kind of finish this, we also have Psilocybe zapoticorum. Yeah, that covers all of it. I keep referring to sections. And just to explain sections in a little bit more detail, sections is an informal taxonomic grouping, and they're essentially groupings defined by microscopic features that were intended to imply relatedness. But the Ramirez-Cruz paper that I mentioned earlier, 2013, showed that microscopic features have been shown to be not useful taxonomically within Psilocybe. So the whole kind of spore size or spore thickness kind of has lost its usefulness in terms of trying to categorise Psilocybe taxonomically. So there's been a bit of a shifting around, and there's been a bit of questioning about how relevant this use of sections actually is. But we're still using it. It still seems to be useful. Now, I showed the figure from the Bradshaw preprint, and I thought I'd try to illustrate how the distribution of the genera looked at a certain point at about 15 million years ago. I tried to colour this, but it may not translate very well. I don't think it does it? Where these different sections have ended up. So the white coloured text is clade 1. So that's like Stuntii. You've got Mexicanae that's turning up in Mexico, Cordysphorae, Zapoticorum in Central America, Hopi that you may not be able to read in the red. It's over there. And then in the southeast kind of Asian area, we've got Stuntii, Cubensae, Carolicens, and Subaruginosae.
Caine, just a quick question. Were the continents all joined 50 million years ago?
No, they weren't. No, and I'll come back to that near the end because it's where the speculation occurs, and we can probably have some questions. But yeah, so that's where the different sections landed about 15 million years ago. And that seemed to be where a lot of these sections kind of diverged from each other. I'm going to introduce barcoding genes. So this is where things get a little technical, and I'm going to try to illustrate this as best I can. But the whole idea behind barcoding genes is rather than using taxonomic features, we can use genetic features. So, you know, like as in the same way that we can tag things in the supermarket and the machines can just read it according to that tag. We've been wanting to be able to classify things using genetics in a similar way. What part of the genetics are specific to that species? And there's been attempts at numerous barcoding genes. And a lot of these all come from ribosomes, which are the kind of the molecular tool that kind of then translates genes into proteins or enzymes. But you have things like the large subunit, which has been really good in terms of getting things to genus, but it doesn't really go, it doesn't have enough resolution essentially for then defining things finer than that. Likewise, the small subunit is the same. It's good to genus. It's okay with some species, but the one that we've landed with is ITS, the internal transcribe spacer. So there's quite a bit of variation within the ITS, perhaps too much, and this is kind of what we've been finding, is that it's good for some genera, but philosophy, it seems to be highly problematic. And there are now things that are turning up in the data that are making this a bit more problematic. RPV1 and 2 seem to be really good, but there's just not enough of these sequences on the internet. The vast majority of the sequences available to look at are ITS. There are other genes, EF1 and TEF, and again, people are doing them, but there's not many, not enough to be able to really dive into the genetics now. So this is kind of how that looks. So you've got your SSU, your LSU, in between you have ITS1 and ITS2, and then the 5.8S. This region is highly conserved, it barely changes. This is what it looks like. All the AT genes and Cs. I have tried to outline this in red where the ITS1 is. There's the 5.8S and the ITS2. So you're looking at about 600 characters. So as I described, what I've been doing for the last year is going through iNaturalist, going through GenBank, which GenBank is the main genetic database that everyone uses. But iNaturalist, people are now putting sequences for their observations onto iNaturalist, and you can access those, download them. So what I've done here is I've taken about 250 sequences and tried to curate them as best I can, and then align them. So over time, the ITS sequence changes. So the idea is to then try to align them so that you can at least map out where those changes have occurred. Most people just do this programmatically, and that has actually caused a few problems because it just matches on GenBank, but nobody actually looks at the alignment properly. They just go, oh yeah, it aligns to ITS. That's fine, so that's that species. But it pays to kind of look at what the alignment is, because sometimes it's maybe mapping to a reverse read, which is not actually ITS, it's just part of LSU. And as I said, LSU is great to genus, but it's not good to species. What I've then gone and done is then rearranged all these sequences into the different clades. And you can kind of see how there's all these large variations between clade 1 and clade 2. And this is ITS1, ITS2. There's a lot more variation in ITS1. ITS2 seems to be more conserved. And 5.8S is highly conserved. There's barely any changes in it at all. I'm going to come over this side. But we can also go down to sections. And I haven't shown this on here because then it would just get messy. But this is section Australis. Section Hopii is here. Section Cordus Borae is there. And section Cubensae. This is section Similanciate, section Zapaticorum here. And this is the subsequent ITS tree that I've built from this. Yeah, it's all going to be completely unreadable, but that's kind of what I've been doing. But I can just illustrate where some of these sections line up. So you've got Similanciate, but what's really interesting is section Stunciae kind of just pops out of section Similanciate here. Mexicanae, Cordus Borae, Zapaticorum. These are all just names, so I'm going to try to put some names to some of these. So section Similanciate are represented by Similanciata, but also Aludicea in Australia. So this is just diving into clade 1. Again, you can kind of see how it's all aligned. What's, I guess, the point behind part of this is also just that the ITS seems to be mirroring the sections really quite well at this point. I need power. And this is section Savuiensis, which is from section Mexicanae. It just looks like my battery's running out. And this is section Papuana, which comes from section Zapaticorum. And this has been found in northern New South Wales and also in Victoria, and it's now in numerous locations in Victoria, which is really interesting to see. Yeah, that's better. Thank you. But a section Zapaticorum that kind of, for me, raises some interesting questions. But I'm going to move on. So this is, section Similanciate. Section Similanciate is really messed up. All the different, it just seems to kind of land all over the place, and it's one of these things where we need a lot more resolution from probably full genome to really get a better understanding of what's going on there. This is another species from section Similanciate. I think these are now being found in New South Wales. This is Angulospora, which is from Taiwan. It's growing everywhere in New Zealand and is now being found in Australia. It typically grows in hardware stores in exotic plants. So clearly some of these plants are now landing here in Australia and bringing their fungal biota with them. This is psilocybe tasmaniana. No, it's not. This is psilocybe eludicea. Sorry about that, wrong picture. But we do have psilocybe, well, we're calling it CF tasmaniana. Yeah, no, that must be a slide from an old presentation. Sorry about that. It's not the Tasmaniana that comes from Tasmania, though. This is a whole new species. We think that the original Tasmaniana was actually just, as described by, I'm having a blank. Well, the person who found it, we now think that they described eludicea. It was Watling. Going back to the ITS tree, and in clade 2 we have Aztecorum, Cubense, Caerulescens, and Sparaginosa. This is the representative from section Cubensis. This is looking a little bit more detailed. And I think the next slide is then looking at Sparaginosa. And this is where things got really, really messy. So as a consequence of the McTaggart paper from last year, we ended up with a whole heap of ITS data from across Australia. And so when we start to look at this whole section, things get really, really messy. And it's probably hard to see on this slide, but we have, things are not lining up. So we have Azurescens and Sparaginosa, for example, and then Alenii, all clustered really closely. I think at some point there's Macarorae, and we have Wiraroa turning up within this tree as well. But because we have so much data, this part of the tree is massive, so I have covered across like 3 different slides here. But yeah, it's a bit washed out. And I think, yeah, it's a mess, and that's kind of my prompt to escape from that for a moment. And this is the main program that I use to do my alignments. These are kind of how I see the sequences. And there is 126 Sparaginosae sequences here. And then if I compact these, I mean, if we're meant to be using barcoding to define a species, then it should all collapse down to one ITS, in theory, or maybe a few, or a handful. But if I collapse this whole thing to get rid of the duplicates, like, yeah, some of these groups are like, they've been combined into a group, say, for example, of 21 different sequences, but we still have all these other ITS sequences. This is not helpful when you're trying to define species and talk about species. It's really, really messy. We have, yeah, and there are things like, there is a Wiraroa ITS that matches Sparaginosa ITS from Tasmania 100%. So what's going on there? Meanwhile, we have just all these other ITSs. Okay, so if ITS is not helpful, it's probably a good idea to look at other barcoding genes. As I think I mentioned earlier, ITS is considered the standard, and there's lots of it. But problematically, NCBI has a lot of genes, but there's a real mixture of varying quality of genes. So to be able to produce that image, I had to delete heaps, like hundreds from, because you can download these in one big file, but then go through and you have to delete the ones that have heaps of data missing or have errors. Fortunately, in the last few years, the quality is really improving, so this picture is going to change, hopefully, in the next 5 to 10 years. And then iNaturalist is, as I said, mostly ITS. RPV2 is becoming more available, and this is one of the trees that I've been able to produce using RPV2. And it matches the sections really quite well. What I found kind of rewarding from the RPV2 is that it has confirmed that things like cyanesins are Seberogenosa. Same with azurescens, it matches with Seberogenosa. And the same with the Lenini. So we seem to be getting a lot better resolution with RPV2. So hopefully more people start using that sequence. So if you're out collecting mushrooms and submitting them to get them sequenced, please request RPV2. A little bit of psilocybin evolution, and then I think question. But yeah, so this was, Sister asked earlier about evolution and what did the world look like at certain points. And so this was the world at 85 million years ago, where all the supercontinents were separating. It was a really warm climate. There was no polar ice cap. There was lots of dinosaurs, and fungi were evolving at this time period. Fungi were evolving to be able to break down lignum, so the white rock fungi were evolving during this time period. And this is kind of an illustration of then how much things kind of evolved over this time. And then you have that major extinction event that happened by the meteorite, the one that killed off all the dinosaurs. You can see like angiosperms, the flowering plants, really evolved quite significantly after that point. Fungi have been kind of slowly just kind of evolving over time, but yeah, they also picked up. This was an interesting time in the world. It was quite warm, and then over time you can see the world has cooled quite a lot. The Paleocene, so 60 million years ago, the continents were separating still. The KPA event happened at 65 million years ago. There was still no polar ice caps, and this is when the first psilocybin were evolving. By 55 million years ago, clades 1 and 2 had already diverged. So, where on this planet did psilocybin evolve? And I guess this is hopefully what, looking at some of these ITS sequences, looking at how they've evolved, looking at their various ecologies, we can start to answer that question. It would be nice to think that maybe psilocybin evolved in Antarctica. It makes sense. Clade 2 kind of moved up to here and evolved maybe in Australia or maybe in India. Not sure about Clade 1. Clade 1 is very, seems to really like grasslands, so not sure. But yeah, they evolved somewhere and at some point spread across various continents and started doing their own thing. And I think and then there's been some other interesting reveals from ITS as well. The Bradshaw preprint from December last year went ahead and renamed, or formally defined, Natalensis AFF is Ochrocentrata. So I'm not sure how many of you have heard of this. But it's also been demonstrated that Ochrocentrata will happily hybridize with cubensis. So, so there's this whole issue around defining what a species is. But someone did the ITS sequences for this hybrid, and this is meant to be cubensis as a species, and this is meant to be Ochrocentrata as a species. But from that one individual, you have an ITS turn up from that individual in the cubensis group, and also an ITS that turns up in the Ochrocentrata group. So mushrooms are carrying numerous ITS sequences, and it seems from the different parent lineages. How does this affect our understanding of species? So we're from here. ITS can only provide so much resolution. I've kind of made that point a few times. We need more sequencing. If you're out foraging for mushrooms or psilocybins and you find interesting things, it would be really good to get them sequenced, to be able to get some genes from them, to help build this picture of their ecology. I mean, as you can see, Sphaeroganosa creates a really complicated picture, and I assume, starting to look at more ITS, this is going to be across the board. So we need more data also to be able to examine these barcode haplotypes, and that's kind of how we're referring to the fact that you've got these two ITS sequences turning up in this one individual. There's also this idea of hybrid swarms, is you get really, really closely related species that are able to hybridize, but then breed back with the parent populations. So you've also got some weird things going on there. Again, what is a species? And I guess part of that's part of this narrative as well. It's defining species boundaries and trying to work out what's going on. Yeah, well, it's question time.
Just before the questions, just to make sure, thank you so much. Just to make sure you've been listening, I've got a short quiz for you all to make sure that you've been listening.
Thank you so much, Caine. Incredible intensity of work and very, very informative. So, yes, so I can hand the mic to you for a question.
It's not related to psychedelics.
Oh.
It's related to the camphor trees we have in our area.
Yes.
Do you know it? It's a tree from China.
Yes.
But it goes mad. So some people are injecting the trees with lion's mane and other edible mushrooms.
Yep.
And it kills the trees rather quickly.
Yep.
Those aren't all good.
Okay.
But I'm just curious what that's going to do to the evolution of the natural moiety in this area.
Oh, okay. Bringing in foreign trees, they usually end up being invasive. So
yeah, the trees are the camphor are
yeah,
invasive.
so it's going to push out your native species. This is the problem with introducing foreign species. Is they push out the native species and overtake those environments. So you get less genetic diversity within the remaining plant populations or the remaining fungi populations.
Well, that's what I'm saying, is that they're injecting the trees with the edible fungi, and I'm concerned what that's going to do to the native fungi.
But then also bringing in native fungi, fungi that are not endemic to an area, they're also going to push out the native fungi as well. So this is a conversation that's kind of starting to emerge widely, is that people be careful what they're growing in their region, introducing oyster mushrooms that don't belong there, or lion's mane that don't belong there. Up until recently, there was a fungi that you weren't meant to grow, and you can't. I think the story is you can't import foreign lion's mane. You're only allowed to grow the local lion's mane. Yeah, because of that issue, because the imported species are going to likely out-compete the native species.
If it's a business, they're doing it with thousands of trees.
Yeah, but it's a push to keep the species that you're growing should really be contained from your local area. Yeah.
Might move on to another question if we could.
Yeah, so if there's a gene that's proving that Aeruginosa and Entheogenesis and one more, I can't remember, are the same species, but they were considered separate earlier because of morphological features, if that's what happened. Yeah. Why is the gene now seen as the thing that's dictating which species it's in? Is that gene that says that those three species are the same now proving that spores from each of those ones can reproduce? Or why is that the reason for it being the same species?
I think the thing is, this Aeruginosa has a really interesting picture because it's so widely distributed. So you have populations from North Queensland down through to Tasmania. So these populations have diverged over time enough that there's genetic differences. So the ITS sequences are different enough. But through looking at other genes, we can go, oh, but this is the one species. The thing with Aeruginosa, Australis, and Caine and Zelenii, they also have quite broad morphological variation. So even within one population, like in a Tasmanian population, you might get different phenotypic appearances. Enough for people to go, yeah, but that looks like a completely different species to that. Part of my point though is ITS is not good enough. There's a lot of conversation on the internet about, well, I've got this ITS sequence that shows that is that species, but perhaps some of these things are actually just part of a species group. So, like the Tasmaniana, for example, that I pointed out, may simply just be Stutzi. It's just appearing with a different morphology. I think that answers your question. Yeah.
All right. Any other questions at this stage?
I think there was a question over here. Did someone put their hand up? Or?
No? Hands up? I guess I could ask, is ITS a barcode for other genera, other fungal genera as well? Yep. And is there the same degree of homogeneity of 5.8S, for example, in the other genera as there is? So that's strictly largely conserved.
5.8 is really high.
It's right across the fungal sort of all of that.
Yeah. So the problem at the moment is trying to identify a more suitable barcoding gene.
Right.
That has enough variation that you can go
differentiate that on.
ITS doesn't seem to do that, and it's causing kind of a bit of a problem because, well, what is it?
Right.
Like if you've got an ITS match that matches opposite strata, it just muddies the water in terms of how we're defining the species.
Right. Any other questions coming up at this stage? I've got another one. You were talking of the psilocybin cluster, so the gene cluster. Do you know of any other genera? I mean, this is a distinctive characterising feature of psilocybin, isn't it?
Yeah, it is.
So, do you know of any other genera that have a combination of any of those within those genes within the cluster, but have lost one or two critical genes for a couple of of those enzymes. So these genes essentially code for enzymes which are involved in the biosynthesis of psilocybin in the mushroom.
Hang on. I might have it in this. I might not have it in this. But you've got...
Yep, that looks promising.
Here we are.
Yep.
So you've got horizontal gene transfer, where bacteria are carrying the gene cluster from psilocybe then into Australis or other groups.
Right.
And here you've got like Jim Pilot's delivers. I think it's got mostly everything.
So I didn't see that one before. So yeah.
This is from a different presentation.
So and in a couple of closely related genera, then you are seeing some combinations of those.
Yeah, but I know in some I think there has been a loss of some of those.
Right. So hence we won't see psilocybin, but there may be some other downstream metabolites. Is that a characteristic of defining metabolites as well?
Possibly.
But we haven't really, nobody's really looked at that yet. So
Yeah.
you might have to speak up a bit. Yeah, the music, so we can somebody ask the music to turn down a little bit? That'd be really unique.
But there is work at looking at other comparative regions in other species. So like looking at, I think like cordycepin, for example, and how perhaps psilocybin might have actually been originally a part of that pathway. That it's just evolved over time. Yeah. Cordycepin's gone off and done its own thing. Yeah.
So genes can evolve out as much as they can evolve in a way. Just Okay, any other further questions before we finish up? I think we're all done. So please join me in thanking Caine Barlow very, very much for this.
And before I finish, I'm going to go right back. I'd like to thank MardiGrass and also EGA for inviting me along. So this is a shared space between EGA and MardiGrass. And I'd like to point out that if you enjoy this talk, if you're enjoying these talks, please come to EGA at the end of the year.